So today I have a guest, a very special guest, Dr. Mandana Arbab. Dr. Mandana Arbab is the Lodish Family Assistant Professor of Neurology at Harvard Medical School and faculty member of the Rosamund Zender Translational Neuroscience Center at Boston Children's Hospital. She received her PhD in Regenerative Medicine at the Hubercht Institute in the Netherlands and completed her postdoctoral fellowship at the Broad Institute with Professor David Liu, where she developed high-throughput CRISPR assays to characterize genome editing tools in mammalian cells and predict genome editing outcomes. She has applied these insights to 3 genetic neurological disorders with genome editing therapeutics in cells and in mice. In 2021, Manda was awarded a Pathway to Independence Award from the NIH National Institute of Neurological Disorders and Stroke. And recently, she's been working on some very exciting projects with base editing, developing and publishing in Science in this year, base editing rescue of spinal muscular atrophy in cells and in mice publication. Manda, it is a pleasure to have you here with me today. Welcome.
It's a pleasure to be here.
All right, so I would like to jump in and to speak a little bit more about the gene editing approaches to spinal muscular atrophy, which of course is your expertise. Let me ask you just to describe the disease. How does it manifest? How does it make sense, again, to target it with gene editing and gene therapy approaches overall?
So spinal muscular atrophy, or SMA, is a degenerative motor neuron disease, which is caused by loss of motor neurons. And these are nerve cells that are located in the spinal cord, and they make contact with skeletal muscle to initiate contraction. And in SMA patients, these neurons are lost, ultimately leading to paralysis, and typically resulting in fatality due to respiratory distress in patients if left untreated. And it is actually the most common genetic cause of infant death worldwide. And most patients, type 1 patients, will develop symptoms between 2 weeks to 6 months after birth. And again, if untreated, will unfortunately succumb to that before about 18 months to 2 years. And this disease the mechanism of it is actually still quite poorly understood, but the genetics of it are very well known. And it all revolves around these genes called SMN genes, which is survival motor neuron is the name of the gene. And this gene, this protein, is actually essential for all cells. But in SMA patients, they suffer from SMN insufficiency, and it's motor neurons specifically that are most sensitive to this insufficiency, and it results in degeneration of those cells. And what is very unique about SMA from a genetics perspective when it comes to gene editing is that the SMN1 gene is lost in these patients. And when I say lost in this case, it's usually completely deleted from the genome. But SMN patients will always retain essentially a backup gene, so to speak, SMN2, which is an almost identical copy, but it has one mutation relative to the good gene, SMN1, which is a C to T nucleotide change. And what's great about that from a genome editing perspective is that means that all patients could also be treated if you can just alter this one nucleotide. So when a lot of even monogenic disorders, you're often grappling with the fact that there are many different sorts of mutations. And so any one genome editing strategy that you would develop might only work for subset of patients. Whereas here, if we could get an effective genome editing therapeutic to market at this target, you now have something that is amenable for all patients of this disease.
That is quite incredible. And of course, we know by now there are at least 2 therapeutics that have been approved on the market, but it has been a long way history towards developing them. Could you speak a little bit about how maybe they were conceptualized, why it made sense in the beginning to target it that way, because the tools were available probably, and because some of the research was done before in the same fashion, but now with the more novel tools that are cleaner, more precise, which of course is something that you will speak about in much more detail, and it's very exciting. There are just much cleaner way to target the same diseases that we targeted previously with AAVs, adeno-associated viral vectors, or the antisense oligonucleotides. So could you please speak to that?
Sure. Yeah, so there's actually, in fact, even 3 drugs on the market for it now that all act at the genetic level, so to speak. The very first drug on the market for this came out, I believe, in 2017. It's called Spinraza, or nusinersen, and this is an antisense oligonucleotide. This drug, as well as the most recently approved drug, risdiplam or Evrysdi, are splicing enhancers. So what I mentioned previously about the one nucleotide change that differentiates SMN2, the insufficient gene, to SMN1, which is the good gene, is that they cause missplicing of exon 7. And exon 7 is the last exon normally of this gene. And there are very many negative regulators of splicing in that region that cause this skipping of this exon. And both the ASO drug and the small molecule, nusinersen and risdiplam, modify that and facilitate splicing inclusion of exon 7 again to basically overcome the limitation that SMN2 genes have to upregulate full-length SMN protein like SMN1 would produce from that gene. And they don't do this entirely to the same efficiency as SMN1 genes would. So we know that in SMA patients in the spinal cord, SMN protein is reduced by about 6-fold relative to normal wild-type unaffected people. And these drugs typically will upregulate SMN expression by about 1.5 to 2-fold in the spinal cord. But this is clearly good enough to, there are many people who are now alive and being treated very functionally by these drugs. But there's a bit of a shortcoming there where, there's potentially room for improvement with a new drug. And then, as you alluded to, there's also an AAV gene therapy called Zolgensma that is available for this disease, which is a self-complementary AAV system that simply has a constitutive promoter overexpressing SMN in whatever tissues are transduced by this drug. Again, also upregulating SMN levels in the spinal cord by about 1.5 to 2-fold and having quite good outcomes.
So that's fantastic, but let's speak a little bit more to the limitations of those approaches, kind of like priming our conversation about the base editors and even cleaner approaches. So with the ASOs, what are the limitations perhaps in the delivery mechanism or the way in which the patients are affected by having to redeliver the ASOs, of course? And what are some of the limitations of the systemic delivery of the AAV9 gene therapy, like in Zolgensma?
Yeah, so the ASO drug has a pretty long half-life of about 3 to 4 months, but the delivery is intrathecal, and so patients have to undergo these, somewhat invasive and, technically require some expertise to perform a couple times a year for presumably their entire lives if they were only to stay on this drug. And so that's a bit of a limitation for patients. Obviously, it doesn't allow for the total freedom, that we all enjoy. And then the small molecule that's on the market does solve some of those issues. It needs to be taken daily, but it's orally available. That one has been on the market most recently, so there isn't as much long-term efficacy data, but it does seem to be doing quite well, but still does need to be taken over and over again. Zolgensma, on the other hand, the AAV drug, may be a permanent treatment. This is something that we will have to see over time, whether or not it does really give sustained expression. To a certain degree, it may well, because our target tissue here is a neuron, which does not divide, so the genome, AAV genome, could stay present long enough. But there are also some thoughts that it might silence over time. It is a viral genome after all, and our cells kind of are resistant to that to a certain degree. And it is becoming a little apparent now in patients that doctors are treating, that patients who are treated with combinations of these therapies, so for example, patients who had received Zolgensma already and are also being given Spinraza on top of that, actually appear to have better outcomes than those who are treated with these drugs alone. That indicates already that there is a bit of a gap in the level of rescue that any one of these drugs alone can achieve in patients. And part of that may well be due to the fact that none of them are really rescuing native levels of expression or allowing for endogenous levels of regulation of SMN expression to the degree that it probably should occur. And these are all areas, I think, that a genome editing approach, in particular a base editing approach to make this very precise change to convert these insufficient SMN2 genes into these good SMN1 genes, could really come through to provide more of a benefit, where you could have a one-and-done therapy, so you don't need to have continuous administration for your whole life, but also recapitulating native expression levels and maintaining endogenous regulation of gene expression.
So that's wonderful. So this is a great segue to speak about the base editing approaches. So tell us a little bit more about how was it conceptualized, how was the mutation targeted, and how did you think about the delivery, kind of like the strategy with this gene editing approach, also thinking maybe long term, of course, taking it from mice to the clinic and to FDA approval?
So the target that we wanted to go after, which is the one that we ultimately succeeded in and continued with our mouse studies, was kind of like the perfect one that we had always wanted to, which is to change this one C to T change that differentiates SMN2 from SMN1 genes, back again. For a very long time, we weren't able to do that. The right tools didn't exist for the job. There were many other strategies actually that we pursued that were also successful at upregulating SMN expression, and all of this rested very heavily on this beautiful, massive body of data that existed from prior research. A lot of it was actually generated in the process of finding the ASO therapeutic that's currently on the market, that identified all of these other negative regulators of gene expression that we found genome editing ways to modify, either using nucleases or base editing strategies. But in the end, we found magic base editing strategy that really was very efficient at converting the one nucleotide that we were after. It's using an enzyme, an adenine base editor enzyme, AB8E, that was also developed in David Liu's lab where I did my postdoc. And we basically screened all of these strategies in a cell culture model, which were mouse CS cells that were derived from the Elsman delta 7 mouse. And this mouse has been instrumental both to our work to study this disease, but actually to the development of, I believe, all of the currently FDA-approved drugs for SMA. It is a mouse model that is pretty good at recapitulating the phenotypes that occur in humans. And most importantly, for a genome editing approach like what we're after, is the fact that it contains the full-length human SMN2 gene. And that is, incredibly powerful if you're trying to develop a drug to treat a human disease, to also be able to have that human gene in the mouse model. And so we had basically also, because of the other drugs that had been developed, a pretty clear path with, there was a lot of data on endpoints and expected phenotypes that we could compare to, and a lot of prior data on also delivery modalities. For example, for Zolgensma, they use AAV9, so we already knew that this is a delivery modality that will target the right tissues, can do so in the correct time frame, and even works in people. So we really were able to stand on the shoulders of those who came before us and use a lot of that information to our advantage.
So that makes perfect sense. So you had, again, an incredible body of data and experience to come up with this strategy, with this approach. But for our listeners, let me just, explain a little bit of the differences again with some of the AAV9 approaches of delivering genes, such as in Zolgensma, where again the virus is having a load, it's delivering the whole gene right into the tissue, spreading it throughout the intravenous delivery into all of the cells, but the virus is very specifically infecting the neurons and the spinal cord neurons, motor neurons, in order to express the gene that is missing. So this is one type of delivery in which the DNA then exists in the shape and the form of the episome, so it sits next to the DNA, does not integrate into the human DNA in the nucleus, but it sits kind of like a circle right next to it, and it is expressed continuously from it. Versus the strategy with the CRISPR gene editing approach, where we go into the gene that's already there in the nucleus of the cell, and in a very clean manner with base editing, we're swapping one letter into another, right? So Let's talk a little bit more about this. And I know you already mentioned the epigenetic silencing that happens potentially with...
May happen, yeah.
May happen, yes, we don't know if it happens. May happen with, again, the AAV9 ways of delivery of the gene product. So what about the gene editing? Is there any reason to believe that somehow modifying the native DNA could lead to any epigenetic changes?
It wouldn't be inconceivable to think so, but from all of the information that we have to date on doing genome editing at these sorts of loci, that doesn't happen. That, you certainly could implement CRISPR genome editing as a modality to make a change that potentially would actively, intentionally alter an epigenetic state. But the presence of CRISPR itself in making a genome edit somewhere does not appear to inherently carry that as a particular risk. An important point to CRISPR genome editing is the fact that with whichever modality you are doing CRISPR genome editing, typically you end up altering your target site. So you load your CRISPR system with a guide RNA that's specific for a particular genomic locus, and then once there, it makes an edit. By doing so, typically it also changes that target site such that it no longer actually recognizes that target site the way that it did before. So the CRISPR itself acts as a one and done at the on-target locus effectively anyway. So it also doesn't necessarily linger in that same position for an extended period of time to our understanding. So that would also make it less likely for this sort of phenotype to occur.
And then just out of curiosity, so when you deliver CRISPR, so actually the Cas9, let's explain this a little bit more, again, for a more general audience. The Cas9 comes inside of the AAV9 vector, and it's actually split in half because it couldn't fit into one vector, right? So then is Cas9 being constantly expressed in those cells that have already been, again, modified and targeted? Does it happen all the way throughout the life? Will there ever be immunity that is developing towards, again, this Cas9 constituous expression?
Yeah, this very much depends on the delivery modality that you choose. So there are, for different applications of CRISPR that people are pursuing for the clinic, there are different answers to that question. So there are approaches that use, for example, RMP delivery, which means that you deliver just the protein, which degrades pretty quickly. Mm-hmm. There's mRNA delivery, which has a little bit longer sustained expression. And then AAV delivery, which is what we're using, would indeed, as you're suggesting, result in long-term expression of this transgene, of this CRISPR system, which arguably is something that is not so desirable. And that definitely is something that is actively being worked on, is can we build into our AAV delivery systems also a mechanism where you don't have constitutive expression of these genome editing tools for your whole life? Because, there may be silencing, as you said, from the AAV genomes over time, but we don't know that for fact, and we don't know that it is complete. And ultimately, the whole idea of these therapies is that they can be one and done, so they should go away. There's no reason for them to stick around any longer. So at present, the system that we have used for our strategy for SMA did not have any of these regulatory systems built in that stop expression, but hopefully, by the time we get to the clinic, we and other people who are all actively working on this topic would find also a way to limit temporally the expression of these genome editing tools.
So that's a great point. Of course, up till now, the gene therapies are known to be those irreversible treatments, but there's some work being done to make them potentially either reversible in some shape or form. Or tell us a little bit about what's been new or what's been in the works in that field.
So for genome editing, the whole idea there is that really you don't want it to be reversible. You wouldn't do genome editing if you don't want it to be reversible. You want to take something that you know is like a real problem and have a permanent fix for it. But there are also even CRISPR-based approaches that are being pursued for gene-based therapies that are RNA targeting, for example, and these might also only have a transient effect and might be able to regulate them differently.
Yes, that makes perfect sense. And of course, we want to correct the gene once and never come back to it again, and want to correct the clinical phenotype. But if we speak about the potential off-target effects that are not known yet, right, or long-term effects, so this is kind of, I think, where the field is thinking to potentially make it reversible to in some way to know how we can kind of like take it back or edit it again in a way that it wouldn't have the off-targets.
Potentially. I think trying to pursue off targets by reversing them again might be a complicated situation because you don't know how many you have. I think for the most part, the best strategy is to try to, you know, go with approaches that seem to have very limited, if any, below detection off-target effects in the first place. And then again, trying to limit temporally the expression of these genome editing tools in the first place, because that would be the most significant way that you could prevent off-targets from happening, is to only have a short burst of them being present in the first place. Hopefully, they would predominantly just edit the on-target and have very little effect on potential off-targets, and then have them go away.
Mm-hmm. Makes sense. So tell us a little bit more about the exciting part of the mouse phenotype that you've published upon, because it seemed to, in very well manner, to reverse the disease phenotype, again, the ability to improve locomotion in mice, and you can describe us just a little bit more about that.
Yeah, so we, as you mentioned, developed a split base editor system. So our base editor is too large to fit in a single AAV, but we split it into two different ones that can come together if they co-transduce, if they target the same cell, which they do quite efficiently. And so we gave this to these SMA mice when they were newborns on day 0. So similar to human patients, where typically the disease onsets at about 2 weeks to 6 months after birth. In SMA mice, it onsets pretty immediately, and they begin to have notable phenotypes at about 5 days after birth. And so you really want to be treating them as early as you can to correct SMN expressions before those motor neurons are irreversibly lost. So we treat them on P0, and then we looked at a number of different outcomes in these mice. So typically their median survival, if untreated, is about 17 days, as it was in our hands. And by around P7, already you can start doing certain motor assays, like writing reflex assays, to see what their motor development is like, as well as doing electrophysiological measurements of the motor neuron function. And we saw improvements in these sorts of assays as well after treatment, as well as a 33% extension in life expectancy, both in experiments done in our facility as well as another facility with the group of Arthur Burgess over in Ohio, where their mice had slightly different birth weights, slightly different median survival, but again, a 33% increase in survival. So that was really exciting to see that reproducible benefit to those animals.
That's fantastic, and congratulations, incredible results. I have another question. So if I'm not mistaken, this was intrathecal delivery, right? So the AV9 carrying the Cas9 base editors have been delivered into the spinal cord and then distributed towards the motor neurons. Can you speak a little bit about the biodistribution, what you have seen, and how would you see this being translated into human? Would you target the spinal cord as well in a human? Or would it be just one injection, or does it need to be distributed in any way to make sure that the neurons are targeted and that it actually travels towards, again, the necessary, the nucleus of the cell where it needs to work?
So yes, when we did the treatment in mice, we did ICV injection. So it's not quite intrathecal. Intrathecal would be very difficult to do in a very, very small newborn. But it's basically doing it in the cerebral ventricles, which then allows the AAV to travel through to the spinal cord and target the cells there. The biodistribution throughout the CNS has also been extremely well described already for Zolgensma, which is the AAV gene therapy that uses the same capsid AAV9. And we know that the transduction of motor neurons, in particular when you inject it in neonates, is very good. And that's great for us because that happens to be the target tissue in our case. It also does transduce other neurons, predominantly neurons, throughout the CNS, in the cortex, and several other brain regions. In our case, any neurons that it does, or any cells really anywhere that it does target, is only a benefit, because the only thing we're doing is doing perfect correction of the insufficient gene into the good gene. So everywhere you can do that is only going to be a positive. But what's also really valuable is we know, again, from the Zolgensma studies that there is, a certain bar of the number of motor neurons that you want to target, which is at least over 20% in this mouse model to have a significant impact in rescue. In our hands, we had over 40%, which is kind of the max that you also get with Zolgensma. So again, we're really able to, leverage so much of this information that's already been gleaned from these other therapeutics to help our study out as well.
Fantastic. So tell me a little bit more about what are the challenges of potentially taking the base editors and these preclinical data into the clinic to have a clinical trial. Is there anything that you predict to be challenging in that regard?
Yeah, I think a great number of things. So, currently there is no approved AAV, base editing therapeutic, in vivo therapeutic that's on the market. So if this were to be the first today, then there would definitely be many hurdles that we'd have to undergo. There is a lot of work being done in this area, and so we may well not be the first, and hopefully we'd be able to learn more from other studies as well. But I think the main focuses would be these off-targets, as we've been speaking about, having ways of feeling very confident that we're not going to have a lot of off-targets, and preferably finding ways to control and limit the number of off-targets that we have. And then in addition to that, when we're thinking of clinical trials, at this point, there are obviously 3 approved drugs on the market, and it's a very severe disease. You couldn't really conceive of doing a clinical trial for a drug that you're starting today without having patients that are already receiving the available standard of care. So we have obviously prior history, natural history data where we know what the disease would be like in patients that are untreated, but we can't really have an untreated population as control, nor would we want to. So one of the other great things that we explored in our study, which I think is actually a huge benefit in this matter, is that we actually co-delivered our genome editing strategy along with one of the currently clinically approved drugs that's on the market, which is the ASO drug nusinersen or Spinraza. And we demonstrated that, not only is there no, as far as we could tell, negative interaction of the ASO drug, but there actually was a beneficial effect of co-delivering each of these drugs. So by providing mice with a small dose of nusinersen alongside our genome editing strategy, we were able to have a very first quick rescue by the ASO drug to sustain motor neurons and have them be in a really good, healthy state for our genome editing strategy to take hold. And I think this is going to be really valuable because you can, look forward to future clinical trials where you could then also have a control arm that are patients who are still receiving, a highly effective standard of care, but then also have our genome editing approach in there as well to hopefully allow these patients to come off of Spinraza and still have the sustained rescue from our drug.
Absolutely. That's a very clever approach in terms of already thinking, again, what will make sense in the design of the clinical trial and what is the situations, the state of affairs we have today, right? So do you think it would be possible at all to combine it with the Zolgensma, with those patients being treated with Zolgensma? And I know you've shared earlier that there are some limitations as to how much of the viral genome can be input into a patient of a particular age, particular body size. Could you speak a little bit more about that?
If combining it with Zolgensma may well have a number of caveats to it. So patients who have already received Zolgensma in the past may well not be eligible for another future AAV9-based therapeutic because there would be prior immunity against AAVs. So that is a potential limitation there unless we develop other ways to overcome that or immune suppression approaches that, to my knowledge, aren't fully formalized yet as being effective. You could, I suppose, conceive of co-administering them at the same time, but I just don't see why that would be a good way of doing it. I think you should, if a base editing approach is approved, I think that would probably be the better way of going about it. Or if you then still want to do Zolgensma, then I think you should do that and probably wouldn't want to do both at the same time because, as you mentioned, there's only a maximum dose of AAV that you can give to a person at the same time, so you'd effectively have to give half a dose of each to make that work.
That's right. So you've alluded about the immune response to, again, somebody who has already been dosed with a viral vector will have probably developing some immune response to that type of vector. I've also heard that there are some people who already have some level of immune response to Cas9. Do you know about that?
I'm not sure that I'm fully aware of that. It may well be, but I don't know about
So
that.
yes, I've heard it in some of the recent talks that, again, some people had exposure, again, just from the bacterial exposure because it's
Uh-huh.
a bacterial origin protein. So curious about that. It's nothing that I've come across in
Right.
my earlier experience.
Yeah, it could be a limitation. One of the, I guess, in a way, upsides of the fact that these drugs would be administered to very young patients is that they typically do not have this prior immunity. And that's why SMA patients that are currently, for example, being treated with Zolgensma typically do not have prior immunity against AAVs, whereas if you're treating older people, they have obviously experienced and been exposed to more things in their life. So my hope would be that for at least the very young patients who would presumably get such a treatment, that would hopefully not be a limitation.
And so, just to conclude our talk, I would like to ask you about again your future perspectives. Where do you see the base editing going? Where do you see the clinical trial again for base editing in SMA happening? Where in the future do you see it? What are the prospects?
I would certainly hope that we could get a safe and effective genome editing therapeutic on the market for SMA. It is, if all of these things, work out as hoped for, SMA is an excellent candidate for a drug like this. It's, a very clear monogenic disease where we know exactly a target that we could fix to get essentially perfect rescue, and that would provide I think a great solution and overcome a lot of the limitations that exist to the already very effective drugs that are on the market today. In addition, I would hope that in doing so, we could learn a lot from the outcomes that these patients have and use that to accelerate the development of other genome editing therapeutics for the many other diseases that are out there. SMA is a rare disease, but it's actually not quite as rare as many of these other diseases that are out there. And it's more difficult, obviously, to do trials when you don't have as big of a patient population. So not only would developing a drug like this be a great benefit, I think, to SMA patients, but hopefully to the broader rare disease community and even beyond that.
And my final question is, what excites you the most again about the field of the gene therapy, the field of CRISPR genome editing as of now?
I'm just generally extremely excited about the field. I think that the speed of the technological development in recent years has been incredible, and with the amount of brilliant people who are working on this still, I don't think it's necessarily at any point of slowing down. And I think there is great promise in being able to do genome editing correction for many monogenic and hopefully even in the future polygenic diseases. So I believe that at some level, this is the future of medicine, and so I hope that I'm excited to be part of a community that's contributing to trying to get us there, hopefully properly and safely in the coming years.
Fantastic, Manda. It's been such a great pleasure to have you on Gene Therapy Insights podcast. Thank you so much. It's been invaluable to hear your opinions, to know your expertise, and to bring it to a very broad audience. Thank you so much for being our guest today.
Thank you. This was great.
Thank you for tuning in to the GTI podcast. I hope you enjoyed the episode and found new insights for yourself. If you like the episode, please press like, subscribe on your favorite platform. Your best support will be sharing this episode with colleagues and friends. For the future content, comment below with suggestions of guests to interview and questions you would like to be answered.